It is 10:15 on a Tuesday night. You are on the couch or in bed or at a desk with a screen in front of you. The show is still running or the phone is still glowing or you are finishing something that became harder to finish 2 hours ago. You are not asleep. You know you should be asleep. You are not going to be asleep for a while. While you are sitting there, something is happening inside your brain that you cannot feel and cannot see and would not believe if someone told you at a dinner party. Your brain is trying to clean itself, not metaphorically, not as a figure of speech for rest or recovery, literally, physically. A fluid-based waste removal system that only operates when you are unconscious is attempting to flush the metabolic debris of 16 hours of waking life out through channels between your neurons. Channels that are currently closed because you are awake, closed because the chemical signature of alertness keeps the cells lining those channels swollen shut. The cleaning system is ready. The channels are sealed, and the longer they stay sealed, the more the waste accumulates. One of the compounds accumulating right now in the spaces between your neurons is a protein called amyloid beta. You have heard of it. It is the protein most strongly associated with Alzheimer's disease. It does not accumulate catastrophically in one night. It accumulates incrementally. Incrementally across nights and weeks and months and years of a brain that did not finish its cleaning cycle because the person it belonged to was not asleep when the cleaning system needed them to be. This is not a story about sleep. Not exactly. It is a story about what your brain is doing right now tonight while you are still awake. And it is a story about a window, a biological window that opens at approximately 10 in the evening for most adults and begins closing after midnight. A window that is not about how many hours you sleep, a window that is about when. The window is open, you are sitting in front of a screen, and the biology does not wait. Before we get to the waste, we have to understand what the brain actually is during wakefulness. Because the brain during the day and the brain during sleep are not the same organ running at different speeds. They are closer to two different operational modes of the same hardware, day mode and night mode. And the night mode cannot run while the day mode is active. During wakefulness, your brain operates in a state of continuous electrochemical expenditure. Every thought, every decision, every sensory perception, every moment of attention is the product of neurons firing. Billions of them, constantly. Firing requires energy. Energy consumption produces waste. The waste of neural activity is not abstract. It is a specific set of chemical byproducts, metabolic debris, that accumulates in the extracellular space, the narrow channels between brain cells as the day progresses. In other organs, waste removal is handled by the lymphatic system. Lymphatic vessels run through your body, collecting cellular waste and returning it to circulation. The brain has no lymphatic vessels, for reasons that have to do with the blood-brain barrier and the structural constraints of tissue housed inside a sealed skull. The brain's lymphatic equivalent was not identified until 2013. It is called the glymphatic system. The name is a portmanteau of glial cells, the support cells that make the system work, and lymphatic. The discovery was published by Maiken Nedergaard and colleagues at the University of Rochester. It fundamentally changed the understanding of what sleep is for. Not what sleep does to the body, what sleep does specifically, mechanically, to the brain, and why it cannot be done any other way. Here is the mechanism. It is specific, it is physical, and it does not respond to intentions. Your brain is bathed in cerebrospinal fluid. The same fluid that fills your spinal column. It circulates through cavities inside the brain and around its surface. During wakefulness, this fluid does not penetrate deeply into the brain tissue. The channels through which it would need to travel, the spaces around the blood vessels that thread through the brain, called the perivascular spaces, are narrow. The cells that line them, astrocytes, are swollen. The norepinephrine that keeps you alert and functional during the day also keeps those cells slightly expanded, slightly pressed against the vessel walls, slightly constricting the channels. When you fall asleep, norepinephrine drops sharply. The astrocytes shrink. The perivascular channels open. Cerebrospinal fluid rushes in. The interstitial space, the space between neurons, expands by approximately 60%. A pressure wave of fluid, driven partly by the pulse of blood through cerebral vessels and partly by synchronized neural oscillations, begins pushing through the expanded channels. Waste products in the interstitial fluid are swept along. They drain into venous blood. They leave the brain. The rate of amyloid beta clearance during sleep is approximately twice what it is during wakefulness. Not 10% better. not marginally improved. Twice. A finding published in Science in 2013 by Lulusier and Nedergaard's group. The brain's primary mechanism for removing the compound most associated with Alzheimer's is only operating at full capacity when you are unconscious. But here is where the body clock enters the picture. And here is where the story stops being about sleep in general and starts being about sleep at the right time. There is a clock in your brain, not a metaphor. An actual molecular oscillator, a cluster of roughly 20,000 neurons sitting directly above the point where your optic nerves cross, in a region called the suprachiasmatic nucleus. These neurons have been tracking the rotation of the Earth since before you were born. Light-sensitive cells in your eyes send signals to this clock every day, calibrating it to the solar cycle. It knows, to within minutes, where you are in the planet's 24-hour rotation. And beginning at approximately 9 in the evening, this clock begins initiating a cascade of events that prepare the brain specifically for its maintenance window. Not for sleep in general, for the deepest, most biologically specific category of sleep that the brain produces. That category is slow-wave sleep. Stage 3, the brain state characterized by high-amplitude delta waves, slow synchronized oscillations sweeping across the cortex at half a cycle to 4 cycles per second. These are the deepest brain waves you produce. They are not produced uniformly throughout the night. They are front-loaded, concentrated in the first 2 sleep cycles, reaching their maximum depth and duration in the first 90 minutes after sleep onset. And the depth of those slow-wave oscillations, the amplitude of those delta waves, the duration of that first deep sleep episode, is not determined only by how tired you are. It is determined by when in the circadian cycle you fall asleep. The circadian clock gates the depth of slow-wave sleep. It opens a window of maximum slow-wave capacity in the early biological night. For most adults on a conventional schedule, that window opens between roughly 10 in the evening and midnight. After midnight, the gate begins closing. The clock's contribution to slow-wave intensity withdraws on its own schedule, independent of how exhausted you feel, independent of how many hours you have been awake. This is where the mechanism becomes specific and where the difference between sleeping at 10 and sleeping at 1 stops being a preference and starts being a different physiological event entirely. The glymphatic system does not operate equally across all sleep stages. It operates preferentially during slow-wave sleep, during the deepest delta wave oscillations. The synchronized firing of millions of neurons during slow-wave sleep does not only produce brain waves visible on an EEG. It produces pressure, a rhythmic compression and expansion of brain tissue coordinated with the heartbeat and the respiratory cycle that drives cerebrospinal fluid through the glymphatic channels. The deeper the slow-wave sleep, the stronger and more regular the oscillations. The stronger the oscillations, the more forceful the fluid pulse. The more forceful the fluid pulse, the more complete the clearance. Shallow, slow-wave sleep produces weaker oscillations. Weaker oscillations produce less fluid movement. Less fluid movement means less waste cleared, more waste remaining when you wake up. Someone who falls asleep at 10s and enters deep, slow-wave sleep by 10:30 is running their brain's waste clearance system at peak efficiency for the next 60 to 90 minutes. The channels are maximally open. The oscillations are maximally deep. The fluid is flowing through. Someone who falls asleep at 1 in the morning enters slow-wave sleep that is measurably shallower. Not because 3 hours more of wakefulness have passed. The adenosine pressure from wakefulness is actually higher at 1 a.m., which would theoretically support deeper sleep. The shallowness is because the circadian gate has partially closed. The clock's contribution to slow-wave depth has been withdrawing for 3 hours. The oscillations are weaker. The fluid moves less efficiently. The waste is less completely cleared. The incomplete clearance is not dramatic, it is incremental. You wake up and you feel slightly less sharp than you expected. Slightly more brain fog than your total hours of sleep would predict. The amyloid beta that should have been cleared last night is still there, slightly more concentrated than it was yesterday morning. Not catastrophically, just incrementally. Just a little more than yesterday. Just a little more than last week. Just a little more than last year. The accumulation does not announce itself. It operates below the threshold of self-detection, and it compounds. Waste clearance is not the only thing that happens during slow-wave sleep. There is a second process operating in parallel that affects your cognitive function the following day in ways you will attribute to anything other than sleep timing. During wakefulness, every experience, everything you learn, every emotional event, every decision strengthens the synaptic connections involved in processing it. Connection strength accumulates across the day. By evening, the neural circuits that have been most active are running at elevated synaptic weights. The connections are sensitized. This is, in a narrow sense, good. It is the substrate of learning and memory formation, but it cannot continue indefinitely. A brain in which all circuits are progressively strengthened eventually reaches a kind of noise ceiling. The signal-to-noise ratio degrades. The system cannot distinguish new information from the residue of old information. It cannot learn efficiently because the baseline is too elevated. The connections need to be reset. The synaptic homeostasis hypothesis, proposed by Giulio Tononi and Chiara Cirelli, holds that slow-wave sleep is the mechanism of that reset. The synchronized delta oscillations mediate a global downscaling of synaptic strength across the cortex. Weak connections are pruned. Strong connections, the ones that represent genuinely important information, are preserved but recalibrated. The system resets to a dynamic range in which new learning is possible tomorrow. The downscaling is proportional to slow-wave intensity. Deeper slow-wave sleep, stronger delta oscillations, more complete downscaling. Shallower slow-wave sleep, the kind produced when the circadian gate has partially closed, less complete downscaling. The brain wakes with higher residual synaptic noise. The signal-to-noise ratio is still degraded. The capacity for learning and attention the following day is genuinely reduced. Not because you are tired, not because you did not sleep long enough, but because the recalibration process did not have the depth of oscillation it required. The mental fog after a late night is not a feeling. It is the subjective experience of a brain that did not finish its reset. There is a regional hierarchy to what slow-wave sleep restores. Not all brain regions benefit equally, and the region most sensitive to incomplete slow-wave sleep is the region responsible for the cognitive functions you most rely on during the day, the prefrontal cortex, the front portion of the frontal lobe, the seat of executive function, impulse control, risk assessment, emotional regulation, and the kind of deliberate reasoning that distinguishes a considered decision from a reactive one. The prefrontal cortex is the last brain region to develop fully during adolescence and the first to show degraded function under conditions of sleep disruption. This is not coincidence. It is the most metabolically expensive region of the brain and the one that accumulates the most operational residue during the waking day. It requires the most thorough overnight restoration, and slow-wave sleep is the restorative mechanism it is most dependent on. Research by Matthew Walker and colleagues showed that even one night of incomplete slow-wave sleep produced a 60% increase in amygdala reactivity, the brain's threat detection center, combined with a measurable reduction in prefrontal control over amygdala responses. The regulatory connection between the prefrontal cortex and the amygdala, the circuit responsible for keeping emotional reactions proportionate and rational, degrades with insufficient slow-wave depth. The result is not obvious impairment. It is subtler than that and more socially consequential. You are slightly more likely to interpret a neutral comment as critical, slightly more likely to send a message you will regret, slightly quicker to conflict and slower to repair it, slightly less able to hold a complex problem in working memory long enough to reason through it, slightly less creative in the way that matters, the kind of creativity that requires holding multiple disparate ideas simultaneously and finding the connection between them. None of these effects is dramatic enough to attribute to sleep timing. You attribute them to stress, to the difficulty of the situation, to other people, to a personality you have come to accept as fixed. The brain does not label the deficit. It simply operates with the prefrontal brake partially released and lets you narrate the consequences however you choose. It is now approaching 11. You have not moved. The screen in your hand has been doing something to your brain's clock for the past hour that you cannot feel, but that has measurable consequences for everything described above. Light-sensitive cells in your retina, called intrinsically photosensitive retinal ganglion cells, IPRGCs, contain a photopigment called melanopsin. Melanopsin is maximally sensitive to light in the blue wavelength range, approximately 480 nanometers. This is also, not coincidentally, the peak emission wavelength of the LED screens that now constitute the primary light source in most people's evening environments. Every photon of blue wavelength light reaching your retina sends a signal to your suprachiasmatic nucleus. The signal says it is still daytime. Stay alert. Delay the nocturnal cascade. The nocturnal cascade that gets delayed is specific. Melatonin, which should be rising steadily in your bloodstream by 9 p.m., is suppressed. Research by Charles Czeisler and colleagues at Harvard demonstrated that blue light exposure at the intensity produced by a tablet or smartphone suppresses melatonin for approximately twice as long as green light of equivalent brightness. Versus green light's 1.5-hour circadian delay, blue light from screens shifts the clock by approximately 3 hours. Your master clock has received over the past hour approximately 60 minutes of continuous daylight signal. It has pushed your melatonin curve later. It has held your core temperature higher than its natural nocturnal trajectory, partially blocking the thermal drop that the sleep-initiating neurons in your hypothalamus require before they will fire. It has elevated alertness slightly, not enough to feel, but enough to extend your natural sleep onset by 45 minutes to an hour. Now here is the compound effect. Your clock has been pushed approximately 3 hours late. The slow-wave gate, which would have opened and been fully accessible from 10 p.m. onward, now does not open until approximately 1 a.m. When you fall asleep at 1:30, the gate is already narrowing. The slow-wave sleep you enter is shallower not only because of the time, but because of the light that moved the clock. The glymphatic system runs at partial capacity. The synaptic downscaling is incomplete. The prefrontal restoration is attenuated. You slept 7.5 hours. You feel like you slept 5. The arithmetic does not explain it. The biology does. If you're over 40, what has just been described applies to you with a multiplier that has nothing to do with your lifestyle. Slow-wave sleep declines naturally across the adult lifespan. Research by Eve Van Cotter showed that between the ages of 25 and 45, the amount of deep slow-wave sleep per night decreases by approximately 60%. By the mid-50s, some adults produce virtually no stage 3 sleep on any given night. The decline is structural. The aging cortex generates fewer high-amplitude delta waves. The synchronization across brain regions that produces deep oscillations weakens. The glymphatic clearance that depends on those oscillations diminishes proportionally. This is the context in which the timing question becomes sharp. A 25-year-old who misses the early night slow-wave window still produces meaningful deep sleep later in the night. The circadian gate has partially closed, but the brain's capacity for delta generation is high enough that the remaining window produces something. A 55-year-old who misses the early night window enters sleep at 1 a.m. with both a partially closed circadian gate and a significantly reduced capacity for slow-wave generation. The two deficits compound. The oscillations are weaker. The glymphatic flush is shallower. The synaptic reset is less complete. For anyone whose slow-wave production has already been reduced by age, the 10 to midnight window is where the diminished supply reaches its maximum concentration. Missing that window does not eliminate slow-wave sleep. It reduces what is already reduced. And the consequences of that reduction, degraded clearance, incomplete prefrontal restoration, attenuated synaptic downscaling, matter more when the baseline is lower. The amyloid beta that accumulates incrementally in a 30-year-old accumulates incrementally in a 55-year-old whose glymphatic system is running at reduced efficiency from reduced slow-wave depth compounded by missed circadian timing. The accumulation rate is not dramatically higher. It is consistently higher. And consistency is the variable that matters over years and decades. The connection between sleep disruption and neurodegeneration is not a hypothesis. It is a mechanistic pathway with converging lines of evidence from multiple disciplines. A study published in Nature Communications by Brendan Lucy and colleagues used polysomnography to measure slow-wave sleep activity in cognitively normal adults and measured amyloid accumulation using PET imaging. Reduced slow-wave activity was associated with increased amyloid deposition in the default mode network, the brain network most implicated in early Alzheimer's pathology. Not total sleep duration, specifically slow-wave activity. A 2021 study in Nature Aging by Joe Weiner and Matthew Walker found that reduced slow-wave sleep in participants in their 40s predicted greater accumulation of both amyloid beta and tau, a second protein marker of Alzheimer's risk measured years later. The relationship held after controlling for total sleep duration, sleep efficiency, and a range of demographic factors. The directionality is important. Alzheimer's disease also disrupts sleep, so the question of whether poor sleep causes the disease or the disease causes poor sleep has been legitimately contested. But the glymphatic research establishes a mechanistic pathway that does not require the disease to already be present. Inadequate slow-wave sleep, specifically slow-wave sleep occurring outside the optimal circadian window, produces impaired clearance of a protein that accumulates in Alzheimer's tissue, in brains that have no other risk factors, in people in their 30s and 40s, on nights when they simply went to bed too late. The cause and effect has not been fully established as causal at the population level. That caveat is real and honest and worth stating. But the mechanistic pathway, inadequate slow-wave depth producing inadequate glymphatic clearance, producing incremental amyloid accumulation, is the strongest candidate currently known for explaining why poor sleep quality, not merely poor sleep quantity, correlates with elevated Alzheimer's risk in every major long-term study that has examined the question. There is one more process happening tonight that you will notice immediately tomorrow and correctly attribute to sleep while completely misunderstanding its mechanism. Memory consolidation during sleep is not passive storage, it is active restructuring. The events of the day, the experiences encoded in hippocampal circuits during waking hours, are not automatically transferred to long-term cortical storage. They require a specific biological process, the coordinated dialogue between the hippocampus and the cortex that occurs during slow-wave sleep. The hippocampus generates memory reactivations, sharp-wave ripples, brief electrical events in which the day's experiences are replayed in compressed form. These reactivations coordinate with the slow oscillations of the cortex and with sleep spindles, brief bursts of high-frequency neural activity, to transfer memory traces from the hippocampus to distributed cortical storage. This process requires slow-wave sleep. It requires the specific neural state that produces delta oscillations, and it is front-loaded into the same early night window that supports glymphatic clearance and synaptic downscaling. Research by Jan Bourne and colleagues, using forced awakening paradigms that selectively disrupted slow-wave sleep while preserving total sleep time, showed that memory consolidation for declarative information, facts and events, and the kind of structured knowledge that work and study require, was specifically dependent on slow-wave sleep duration and intensity, not on total sleep time or REM sleep. A late sleeper who obtains 8 hours from 1 to 9 a.m. is running their memory consolidation process in shallow, slow-wave sleep, with a partially closed circadian gate, with weaker hippocampal cortical dialogue, and with fewer and less powerful sleep spindles than a 10 p.m. sleeper running the same total duration. The information that was learned yesterday is less completely integrated by morning. The connections between new and existing knowledge are less robustly formed. The next day's learning capacity is reduced not only by incomplete synaptic reset, but by the incomplete transfer of what was learned the day before. You will feel this as slightly less sharp, slightly less able to access what you know, slightly less capable in the meeting or the class or the conversation where you needed the full version of your brain. You will attribute it to stress or distraction or the difficulty of the material. The brain does not label the source. If you routinely sleep from 1 to 9, the deficits described here are below the threshold of acute awareness. The brain is adaptive. It calibrates to a new normal. It learns to function at 80% while presenting as 100% to the people around you and mostly to you. The glymphatic clearance runs at partial capacity every night. The amyloid accumulation is incremental. The synaptic noise floor is slightly elevated each morning. The prefrontal restoration is slightly attenuated. The memory consolidation is slightly incomplete. The working memory is slightly reduced. The emotional regulation is slightly loosened. None of these effects feels like brain damage. They feel like Tuesday. They feel like getting older. They feel like how life is. The body does not label the deficit. It simply operates at reduced capacity and lets you construct whatever narrative fits. The dangerous quality of the circadian misalignment is not that the damage is hidden. It is that the damage is so gradual and so multisystemic and so below the self-detection threshold that no single bad morning can be attributed to it. The accumulation has no visible before and after. It has only the invisible slope of a brain that is cleaning itself a little less completely each night and consolidating its memories a little less robustly and restoring its prefrontal control a little less thoroughly, and clearing the proteins most associated with its eventual decline a little less efficiently. The slope is gradual. The compound interest on it is not. It is 10:15. You are still on the couch. Your suprachiasmatic nucleus, the 20,000 neuron clock that has been tracking the Earth's rotation since before you were born, has been signaling the preparation for maintenance for the past hour. Melatonin is rising in your bloodstream. Core temperature is falling. The norepinephrine that keeps the glymphatic channels closed is beginning to withdraw. The astrocytes are preparing to shrink. The perivascular channels are preparing to open. Somewhere in the interstitial spaces between your neurons, the metabolic byproducts of 16 hours of consciousness are waiting. The amyloid beta that was produced during the day's neural activity is in the fluid between cells, waiting for the fluid pulse that will carry it out. The synaptic circuits that have been strengthened by everything you experienced today are waiting for the downscaling oscillations that will recalibrate them for tomorrow. The hippocampal traces of the day's events are waiting for the slow-wave dialogue with the cortex that will consolidate them into long-term memory. The glymphatic system is staged. The slow-wave gate is wide open. The circadian window is fully accessible. The screen in your hand has been sending blue wavelength light to the melanopsin cells in your retinas for the past hour. Each photon has been telling your master clock that it is still day. The clock's nocturnal cascade has been partially suppressed. The window that is open now will begin to narrow after midnight. By 2 a.m. it will be substantially reduced. By 4 a.m. the slow-wave machinery is already beginning its transition toward the lighter, dreaming-dominated architecture of the late night. The waste will not be cleared at equivalent efficiency at 2 a.m. The synaptic reset will not be as complete at 2 a.m. The memory consolidation will not be as thorough at 2 a.m. The prefrontal restoration will not be as full at 2 a.m. Not because you will have slept fewer hours. Because the gate will be narrower, because the clock's contribution to slow-wave depth will have withdrawn, because the brain that goes to sleep at 1 a.m. is not the same operational event as the brain that goes to sleep at 10 p.m., even if both sleep until 8. The biology has been keeping a different ledger than your calendar. The ledger does not record hours. It records depth, it records timing, it records how completely the channels opened and how efficiently the fluid moved and how thoroughly the proteins were cleared and how robustly the memories were consolidated and how fully the prefrontal cortex was restored. The ledger entries do not announce themselves in the morning. They accumulate quietly over years and decades in a brain that is cleaning itself slightly less than it should, slightly less than it could, on a schedule the planet set and the clock keeps, and the screen in your hand has been disrupting for the past hour. The gate is open. The channels are ready. The waste is waiting. Put down the phone. Let the delta waves begin. The brain has been keeping a different set of books than your bedtime.